I am David Redden, and today we are talking about rigor, reproducibility, and transparency with specific focus on statistical designs and scientific rigor. Back in October 2015, there was an announcement given out about upcoming changes with NIH, with the way all of the NIH applications that fall under the R mechanism, the K mechanism, the F mechanisms are going to be reviewed, and there are new scorable criteria that affect all applications. Everything is effective, and every application going in since January 2016 should be addressing each of these 4 criteria that are applicable to the grant itself. 3 of them are not optional. So there are 4. There is one called scientific premise, and scientific premise is much more than a literature review. It's a justification of the scientific premise behind what you're doing. You're justifying your approach, comparing it to previous literature, and looking at your preliminary data to see and justify the assumptions with it and identify the strengths and weaknesses in both the literature and your preliminary data. It's to convince the reviewers that you've really done your homework and that your study is going to be a major advancement into the scientific area that you are researching, and the only way you can do that is to demonstrate that you are very well aware of what's in the literature, what the strengths of that literature happen to be, also where the gaps and also the limitations of the literature exist. Every scientific paper that is published usually has a limitations section indicating where future research is needed. Are you aligning with that? And the same thing goes for your preliminary data. You know, any data set that you have that is preliminary, you need to emphasize its strengths but also acknowledge its weaknesses. The other 3 are biological variables, trying to emphasize that within all experiments we need to allow for both sexes in the experiment. Also, biological variables would be age. These things have to be accounted for in the study. If you're studying human subjects, you would also account for race. Those are examples of biological variables that you must account for. And if you're not covering both sexes or all races or all age groups, there needs to be some justification provided. The other one is authentication of chemical resources, cell lines. That is a separate document. It is optional depending on the type of study you're doing. If you're not doing one, if you're doing a study that does not require any of the material, you still have to upload the document, but you say does not apply. And the last one, which is the one that I'm most interested in that we're going to focus on here, is scientific rigor. And it's important to note that scientific rigor actually has 2 definitions. There is rigor with regard to being able to give a step-by-step process by which the experiment is repeated. You would expect individuals to come to the same type of results. And then there is statistical rigor, where you lay out a statistical design that's going to lead to unbiased results, and you will have a system by which all the analyses can be documented, and if that data set is placed in an independent methodologist's hand, they should be able to conduct the same analysis and reach the same conclusions. The reviewers are going to have to come in and find these sections, and they will end up being they will have received instructions that with regard to scientific premise, it must fall in the significance section. With regard to biological variables and with regard to scientific rigor, it will have to fall in the approach section, and they are looking for these things as they are reviewing, and they are scorable criteria. Well, especially with regard to analyses, when we've completed the experiment and we have the data in hand, there are certain things that happen. And there's a great example given in a Nature article where they talk about the Texas sharpshooter approach, in the sense that what you do is you study the data so long and so hard that you actually start seeing patterns where there aren't really patterns within the data. And that leads to the concept of just so storytelling, that after you have seen it, that you start crafting a story that fits what you're observing. And then there's also the idea of sometimes you're myopic in your approach to it. You're so convinced that your model is correct or your hypothesis is correct that you're not asking yourselves, are there other explanations that may do a better job of explaining the variability in the data? Or is there another statistical model that may explain the variability within the data? And the last one, which is one I have the most experience with in my career, is actually asymmetric attention, where you actually come in and when you're doing the analyses, if you achieve statistical significance, you're more ready to accept that, and you stop digging and you stop looking at different possible explanations. Whereas if you have something that's non-significant, then you start digging and pressing much harder, and you're asking yourself, well, why were my expectations not met? Should I try a different model? Should I look and see if there are more influential points or outliers, to see possibly is there an explanation for why I did not observe what I wanted to see. The way you avoid those is very simplistic in a lot of ways, and these are the strategies that we like to employ. First we want to sit down and really write out the approach to begin with. Be as clear as you can. This is actually a reviewable criteria. It's been there in NIH for years, where you provide a detailed analysis approach, a strategy. How are you going to go about going through the data, and what models are you going to build? How are you going to check the assumptions? What is your strategy to find outliers? And if outliers are there, how are you going to do a sensitivity analysis with regard to those outliers, instead of potentially removing them from the data set? What are the comprehensive strategies? Reaching out to either a colleague, or even better yet, reaching out to an expert in the field that is actually approaching the same scientific problem but from a different paradigm. Let them see the data. Let them be able to say, if I was analyzing this data, or if I had this data in hand, this is the way I would interpret it. And that way to actually understand, is there a different or possibly a better explanation of what you're observing? My favorite one of all of them is the blinded analysis, especially from a statistician's standpoint, to be able to hand a data set that has removed instead of revealing this is drug A and this is placebo, just label them A and B. That way no one's biased in trying to believe which way the p-value should go, or which way what the statistics should look like, and just say, can you tell me if there's a difference between the two groups? And allowing them to do the analysis completely independent of you, to see if they're coming to the same conclusion about the assumptions, to see if they're coming to the same conclusions about which test is best to apply. See if they're identifying the same outliers as you. And therefore, a blinded data set approach is actually one of my favorites. The final one is what we call devil's advocacy, and that is where we come in and literally just say, what is another model that could work, and say, is that superior? For statistical reproducibility, I'm a huge fan of any software that allows you to record the syntax or the code. It is essential that whenever an abstract is going out, or whenever a paper is going out, or anything that is going to be documented from a data set, the data set itself must be archived, but also the procedures that produced the results that are reported. I'm always concerned about graphical user interfaces, or GUIs, where you're pointing and clicking, because after that abstract goes, or after the paper is submitted, there's really no trail of breadcrumbs showing you exactly what has happened. Whereas if you've written syntax, or if you've written statistical code, whether that's in SPSS, SAS, or R, all of that can be archived along with the data set, dating the abstract, dating the paper, and therefore if questions come back from reviewers or someone else that says, hey, what about this? You have immediate access to exactly what you did and the data set. It's also very good because down the road, people may reach out to you and say, hey, can I have the data set from this publication, and can you share your code with us? And it's good to always be able to place your fingers exactly on materials that produce those results. I'm a huge fan of the BIRD resources that are available throughout the partner network with regard to being able to reach a set of methodologists throughout the network. Tulane, South Alabama, University of Medical College of Mississippi. All of us, there are statisticians and methodologists stationed throughout the partner network and have office hours and walk-in times where you can come in and discuss everything from power to reproducibility to archiving, all the key things that you need to be able to do. Not only can they do this for you with regard to papers and manuscripts, they can assist you in the writing of your grants to emphasize each of the 4 components and point you to resources on the CCTS webpage that can help you with all the 4. But the methodologists are there definitely to help you with scientific rigor, especially with regard to the stats design, power, and data management.